pH is a scientific measurement used to identify solutions as acidic, neutral, or basic. Measuring the relative amount of free hydrogen and hydroxide ions in a solution identifies if a solution is acidic, having a greater concentration of hydrogen ions than hydroxide ions, basic, having a greater concentration of hydroxide ions than hydrogen ions, or neutral, having the same concentration of hydrogen and hydroxide ions. A meter, pH paper, or performing a titration can measure the concentration of these ions. Testing pH with a pen or bench meter offers many advantages compared to testing with pH paper or titration. These advantages include more precise and accurate results. Meters can detect pH values with great accuracy. The meters in this video measure with an accuracy of plus or minus 0.05 pH. Reduced human error. Digital numerical values reduce human error as they do not rely on the human eye to register and interpret color. Reusability. With proper care and calibration, meters last for years. Speed and convenience. Meters provide results in seconds. Pen meters are handheld, portable, and a great choice for taking measurements in the field or lab. Proper care and calibration of pH meters are required to ensure accurate readings. This video will review common best practices for pen meter and bench meter use. Always read and refer to the instruction manual for operation, maintenance, and storage directions that are specific to your pH meter. When using a pH pen meter for the first time, condition the pH electrode by immersing it into pH electrode storage solution for 1 hour before using. If the electrode protection cap is dry, add storage solution in the cap and soak the pH electrode for 1 hour. Your pen meter is now ready for first-time use. Before and after using a pen meter to test a solution, rinse the pH electrodes with pure distilled water and wipe clean. Many modern pH meters are factory calibrated and do not require initial calibration. However, over time, many models do require periodic calibration to ensure accuracy. You may wish to calibrate your meter after extended storage or when extreme accuracy is important. There are 3 different types of calibration when it comes to pH meters: single point, 2-point, and multi-point calibration. These names refer to the number of points or pH values at which a calibration is performed. The greater number of calibration points, the greater the accuracy of the calibration. These types of calibration are conducted using buffers of known pH values. Refer to the instruction manual for your pH meter for the buffer solutions that you should use. The meters in this video have automatic temperature compensation. This means that the meter automatically corrects for the temperature of the solution. For example, if the buffer solution's temperature is 15 degrees Celsius, the calibration value you will get should be a pH of 4.0 with a buffer of 4.01, a pH of 7.04 with a buffer of 7.0, and a pH of 10.12 with a buffer of 10.01. This video demonstrates the 3-point calibration of the OHouse ST20 pen meter. To calibrate, you will need the pen meter, distilled water, a soft clean cloth, a pH 4.01 buffer solution, a pH 7.0 buffer solution, and a pH 10.01 buffer solution. Before use, remove the protective cap. Rinse the pH electrode with pure distilled water and wipe clean. Be careful not to scratch the bulb of the pH electrode. Press the on-off button to turn the pen on. Dip the electrode into a pH 7.0 buffer. Press the Cal button to enter calibration mode. The pH will be shown with Cal blinking. Due to the automatic temperature compensation, the pH of the buffer shows at 7.01 because the temperature was 22.9 degrees Celsius. The meter will judge if the signal is stable automatically, and once stable, the meter will change to display a value of approximately 4.01, the next buffer for use. The calibration point has been collected. Now rinse the electrode with distilled water. Repeat this process with the 4.01 and 10.01 buffer solutions. Recall that this is a 3-point calibration. Place the pen meter into the 4.01 buffer solution and wait until Cal begins to blink. Once Cal stops blinking, the display will change to approximately 10.01, indicating that the meter has been calibrated for 4.01. Remove the probe, clean the electrode with distilled water and insert it into the 10.01 pH buffer solution. Wait for Cal to start blinking and stop blinking. Upon completion, the Cal will disappear. Note, again, in this case, because the temperature was 22.5 degrees Celsius, the pH of the buffer is 10.03. Due to auto temperature compensation, the pH display shows 10.03. Rinse off the pen meter one final time with distilled water and wipe clean. The pen meter is now calibrated and ready for use. If you are not going to immediately use your pen meter, replace the cap. To use a pH pen meter, first remove the protective cap. Then press the on-off button. Dip the electrode 2 to 3 centimeters into the solution so the bulb of the electrode is covered. Wait until the reading is stabilized. Record the reading. Rinse the electrode with distilled water and wipe clean. Repeat this process as needed to take all your required readings. Once finished, press and hold the on-off button to turn off the meter. When testing pH in the lab, many teachers and scientists prefer bench meters to pen meters. Bench meters often have large digital displays separate from the probe and a flexible electrode stabilizing arm for easy movement and use between solutions. Some bench meters offer greater accuracy or resolution than pen meters. Just like pen meters, bench meters also require proper care and calibration to ensure accurate performance. We will review common best practices for bench meter use. The pH electrode should always be stored in a storage bottle containing electrode storage solution. This prevents the bulb of the pH meter from drying out and damaging the pH meter. Always keep the electrode immersed in its storage solution. Before and after using a bench meter to test a solution, rinse the pH electrodes with pure distilled water and wipe clean to prevent leftover solutions from causing inaccurate results. Like pen meters, bench meters also require periodic calibration to ensure accurate readings. Calibration methods vary between models, and the manufacturer's manual should be referred to for the model of bench meter being used. As an example, we will demonstrate how to calibrate the AquaSearcher AB23pH O-House bench meter, a standard pH bench meter. To calibrate, you will need the bench meter with pH electrode, distilled water, and a buffer solution. This meter uses either a 1-point or 2-point calibration. In this video, we will show you a 1-point calibration. We will use a pH 7.0 buffer solution. First, ensure the meter is plugged in. The electrode is properly attached, and the meter is powered on. Place the pH electrode in the prepared solution. Stir for 4 seconds, and wait an additional 30 to 60 seconds. Press the Cal button. Cal displays on the screen and blinks. A measurement icon will also appear on the top of the screen. During calibration, the measurement icon blinks. When the reading does not change for 5 seconds, the reading is stable. Press the Read Enter button to lock the reading. The bench meter is now calibrated and is ready to be used. Recall that this is a 1-point calibration. Other bench meters may require 2- or 3-point calibrations. To perform these calibrations, repeat the calibration process with additional buffer solutions at different known pH values. Follow your bench meter's instruction manual. Once calibrated and the pH electrode has been rinsed with distilled water, the bench meter is now ready to be used. To take a pH measurement, place the electrode in the sample. Stir the solution for 5 seconds, then wait 30 to 60 seconds. Press the Read Enter button, and the measurement icon will appear on the display and blink until the reading is stable. When the reading is stable, press the Read Enter button to lock the reading. The measurement icon will disappear, and the pH value will be displayed on the screen. Record your pH value. Measuring pH using pen and bench meters is a common and essential laboratory skill. By following proper maintenance and use guidelines, pH meters will provide accurate and dependable results for years of laboratory investigations. For more information about O-House products at Carolina, see carolina.com/ohouse.